All right, we're starting a new chapter here. This is chapter 8 on microbial genetics. And that's one of my favorites because I love genetics. All right, your learning objectives for this first part of your notes is to really explain the flow of genetic material through the central dogma of molecular genetics. Now, this is one of the most important concepts that I want you to take away from this chapter, because this is something you're going to see throughout any of your intro biology courses or things like that in college. It really explains how your DNA codes for your traits. We'll be looking at some different genetic vocabulary, kind of a review of some of the things you studied in biology, and then we'll look at DNA replication as it pertains to prokaryotes. So again, when we talk about genetics, it's just the study of heredity, and that includes things like what a gene is, how your genes carry information, how your genes are replicated and passed on, and the expression of the gene, okay? Because that's what gives you your particular characteristics, your traits. Your genome is all the genetic information in a cell. So I like to sometimes call it your gene home. Get it? Genome? Genome? It kind of sounds like. Now, most of that's going to be on the chromosomes. So in a bacterial cell, it just has that one big circular chromosome. But remember, if you remember when we talked about prokaryotic structure way back in chapter 4, they also have plasmids, which are tiny circular pieces of DNA that can hold like genes for antibiotic resistance to make that particular microbe resistant to certain antibiotics. In our cells, eukaryotes, in addition to our chromosomes, we have DNA in our mitochondria. Plant cells have additional DNA in their chloroplasts. So genome includes all of that. Again, your chromosomes are just structures that contain DNA. They're usually wound up tightly, associated sometimes with certain proteins, like in our bodies, they're wrapped around histone proteins. Genes, though, this is a big one right here, because this is what we'll be talking about a lot over and over again. These are segments of DNA that code for proteins. They call it a functional product, but we're really talking about proteins. So all the different traits you have in your body are because certain proteins were made or were not made in some cases. A quick review of the structure of DNA. Remember, DNA stands for deoxyribonucleic acid. So it's DNA, okay? So it's made up of a whole bunch of nucleotides. Those nucleotides are made up of a sugar, a phosphate, and a nitrogen base. Now, because this is deoxyribonucleic acid, the sugar is deoxyribose, which is a 5-carbon sugar. Your different bases, remember, are adenine, thymine, cytosine, and guanine. A, T, C, and G, we abbreviate that. And so you'll see these repeating structures, like here's a nucleotide, here's a nucleotide, here's one, and here's another one. And on the opposite side, here's one, here's one, here's one, and here's another one. So that's what DNA is these repeating nucleotides that are joined together. And you notice that T always bonds with A, C always bonds with G. These two strands of nucleotides, because it's a double helix, are twisted. That's what gives them that helix shape. So we have all these nucleotides that are bonded together, and then they're twisted to form this double helix. The sides are made up of sugar phosphate backbones. If you looked at the edges here, here's the 5-carbon sugar, and here's the phosphate group. You can see that because there's the phosphorus in there. Okay? So it goes sugar, phosphate, sugar, phosphate, sugar, phosphate. So that's what we call the sugar phosphate backbone. The bases in the in the middle pair up. A with T, C with G. We call that complementary base pairing. And those bases are held together by weak hydrogen bonds. Now, why is it important that those bonds are weak? Remember, we're eventually going to have to replicate the DNA. And later on, we're going to talk about transcription. That means you have to be able to separate those two strands of nucleotides. So by having those bonds be fairly weak, it's a little bit easier for us to separate the strands so we can copy off the DNA a little bit easier. So the structure of the DNA is really what helps explain how it stores our genetic information and how it's fairly easy to copy that genetic information. The sequence of bases carries the genetic code, and that's going to determine what proteins are made. All right? So the order of those bases is really what holds the key to eventually what proteins are going to be made in your body. That complementary nature of those bases also allows for the duplication of DNA during cell division. Because when Watson and Crick were first discovering DNA, they basically said that the genetic material had to be able to do two things. Whatever the hereditary material was in people, in cells, it had to be able to do two things. It had to hold the code for the function of the rest of the cell, and it had to have some way of replicating itself so it could keep getting passed on to other cells. And these 2 points right here just support that. When we look at cellular metabolism, we already covered that a couple chapters ago. That deals with translating that genetic code into specific proteins, and when those get made, we say the gene has been expressed. So a lot of times we say gene expression equals transcription and translation, because we're taking that code from the DNA and we're using it to make a protein so that then that trait can be expressed. Now here's a little review from biology. Genotype and phenotype. Genotype is the genetic makeup of the organism. It's the actual allele combination. Oops. Let's spell that correctly. Here we try this again. Allele combination. Now, an allele is an alternate form of a gene. So if you inherited like a big T or a little t, those are alternate forms. Let's say they coded for height. So if you got, you could either inherit this from your mom or dad, or you could inherit this from your mom and dad. Now, your actual genotype, though, is the allele combination from both of your parents. So you could have, oops, sorry about that. Ooh, I don't know what I bumped there, but anyway, so you could either inherit two big T's, a big T and a little t, or a little t, depending on what alleles your parents had that were in the egg and sperm cell that joined together to form you. So these are what we call our genotypes. It's your actual genetic combination. Your phenotype is the expression of the genotype. So in fact, I like to say it's the physical expression of the genotype, because the pH and the pH right here, okay? So that's the actual trait, like, are you tall? Do you have brown eyes? Those are all phenotypes. So what does your trait look like, basically? Okay, but they can be enzymes. A lot of times we like to think of our outward physical traits that we can see. Well, those are the easiest phenotypes to see. But a lot of it is internal. When you think of all the different proteins that are in your body, and a lot of them are enzymes, that's also a phenotype, whether or not that enzyme functions properly based on the genotype you inherited. So it could be structural, like having certain structures that you can see, or it can be enzymatic, where it helps in catalyzing certain reactions. Now, just a reminder that bacteria have a single circular chromosome, as opposed to we have, 23 pairs of linear chromosomes. Bacteria only have one single circular chromosome, and that can be associated with different proteins. It's looped and folded, and it's attached to the plasma membrane at different points. But remember, we say that it's in that region called the nucleoid. So it's just in that region in the cytoplasm. It is supercoiled and twisted up because we don't want any damage to come to it, just like we don't want any damage to come to our chromosomes either. But so that will play a role when we're talking about DNA replication. There are enzymes that help unwind that DNA. And then genomics is the sequencing and molecular, kind of characterizing the different genomes of different organisms. So here's where they've sequenced a genetic map of E. coli, and they've used little color-coded things there to show where proteins for carbohydrate metabolism and amino acid metabolism are found on this singular circular chromosome. Now again, when we talk about the central dogma of molecular biology, it's explaining how your DNA codes for traits or for your proteins. It's the directional flow of genetic information. Notice that those arrows are only going one direction. So we start out with DNA, and then we go to RNA, and then we go to proteins. Okay, that's the basis of the central dogma. It's how your DNA codes for proteins or your traits. Now, there's three main processes involved that we'll be talking about in this chapter. The first one is replication. That's why we have this arrow kind of pointing on itself for DNA, because that's showing that DNA can replicate itself. When we copy DNA onto RNA, we use the process called transcription. And then when we use the code on RNA to make a protein, we call that process translation. So this whole schematic here is what we call the central dogma of molecular biology. And this just kind of shows you, if you have your DNA here in the parent cell, it can make a copy of that through DNA replication, and then the cell can divide through binary fission and pass that on to their offspring. Sometimes through conjugation or other methods, we can have genetic recombination by transferring some of the genetic information from one cell to another. And then, of course, that through transcription translation, that the information that's on this chromosome can then be translated to make a particular protein. So DNA replication, simply DNA makes a copy of itself, and it has to do that every time a cell is getting ready to divide. Okay? Every time a cell is getting ready to divide, because if that cell doesn't have DNA, it won't have the genetic instructions for how to function. So one strand of DNA acts as a template for the other strand. And the place where replication starts is what we call the replication fork. And there's a lot of enzymes, like I said, that play a role. These are only a few. There's a lot more. And in fact, you have a whole big chart in your book. Gyrase is what helps to relax the supercoiling. Okay. Helicase, you might remember that from biology, unwinds and separates the DNA strands. So it's basically breaking those weak hydrogen bonds. This is the big daddy right here. DNA polymerase adds the complementary nucleotides to the exposed DNA. Remember, A will bond with T and C with G. And then ligase seals up the sugar phosphate backbone to kind of join those. It makes those covalent bonds. It kind of joins that sugar phosphate backbone. So lots of different enzymes involved. We call DNA replication semi-conservative because each new double strand is going to contain one original and one new strand. Okay, one original, which was conserved, and one new strand. Those strands are anti-parallel to each other. That means one strand goes in the 5 to 3' direction, the other one in a 3 to 5' direction. Those 5 and 3' just deal with the carbons in that pentose sugar. DNA polymerase can only move in a 5 to 3' direction. Right? Some bacteria can do a bidirectional replication where they have two replication forks and they can move in opposite directions. In addition to the DNA polymerase putting down those nucleotides, it can also proofread. So usually there's not very many mutations that occur because of the proofreading and double checking. So there you can see the gyrase has come along and relaxed the supercoiling. The helicase has come along and separated these DNA strands right here at the replication fork, and now DNA polymerase can come along and put down these new nucleotides according to the correct base pairing. Okay, again, A with T, C with G. Here's the replication fork. You can tell I'm doing this at school because you just heard the bell ring. All right, because DNA polymerase only goes in a 5' to 3' direction, you'll get one strand that's continuous here, 5' to 3', but then the other strand, because it needs to go in a 5' to 3' direction, it can't just keep going down this way until it has a little piece to start it out. And so an enzyme called primase puts down a small RNA primer, and that's enough to get the DNA polymerase started to start going in that 5' to 3' direction. But it's very discontinuous because that RNA primer, primase will put down little pieces all the way along the DNA to give it a starting point. And so you'll have these short little fragments called Okazaki fragments. So later on, an exonuclease is going to have to come and remove that piece. And when it does so, then the ligase can come in and seal up those openings. Okay, so it's going to come along and seal up any breaks in there. Okay? Form those covalent bonds. Remember, since bacteria cell is one big circular piece here, it's going to it'll have its replication fork, and it's just going to keep replicating until it makes a whole copy. So it's almost like you're making a big 8 here. One from the original strand, and here's the newly copied one, and then when that cell divides into 2, like up here, here's one cell, divides into 2 through binary fission, it now has a complete set of its DNA.